Hello, my name is Albert-László Barabási, and it's a pleasure to be part of this meeting. And it's a pleasure to talk a little bit about our work in my lab about nutrition and what we call the nutritional dark matter. To begin with, I should say that I'm not a nutritionist, nor am I actually a biologist. I'm a physicist by training, and much of my work in the last 20-25 years have focused on the structure of large networks. That is, trying to understand the structure of the internet, structure of social networks, or any type of complex networks that emerge in nature and society and technology, trying to explore the mathematical principles that govern them. It was this journey that about more than 10 years ago has brought me to the question of how does the network within our cells look like, and how does the breakdown of this network or changes in this network correlate and lead to human diseases. And this has been actually a very all-encompassing journey for us, because once we started exploring these subcellular networks, it became the main topic of exploration for my lab. We call this network medicine, and it's a pursuit to understand how the physical interactions between the different proteins and genes and regulatory elements within the cell support cellular function, and then how their breakdown lead to human disease. Network medicine now is a very large subject, and at Harvard Medical School, where I also have an appointment, we now have a network medicine division that has more than 200 researchers exploring the network paradigm in the interest of patients. And what can we do with the network medicine approach? We can identify where the disease lies within these complex networks, that's what we call disease module discovery. We can talk about personalized medicine, that is, how are different patients different, and whether a drug works for one but doesn't work for the other one. We can identify new dark targets as well as repurposing opportunities. We explore dark combinations and so on. And this has been a journey that has been really relying on lots of genetics data, that much of the interactions and much of the starting point of our exploration really relies on genetics. One of the questions we've been asking about 5 years ago is that genetics only offers a limited prediction about the likelihood of a person developing a disease. So we started to ask, why do we die? And how could we really explore, using networks and genetics, this question? And as we started to explore that, we realized that when we look at the top 10 causes of death in the US, which is shown over here, a relatively small fraction of the diseases can be explained by genetic factors only, which raised the question, where is the rest? That is, if only 10 to 20% of the disease occurrence can be explained by your genes and the mutations on them, where is the remaining 80, 90, 70%? What is responsible for that? And it was this question that really brought us to the question of nutrition, because often the answer is, well, it's in the environment. The missing piece is always in the environment, how we interact with that. And one of the strongest components in the way we humans interact with our environment is obviously nutrition. Not the only one, but one of the strongest ones. So we set out to somehow build the concept of nutrition into our models. And of course, I don't have to convince this particular audience how important role nutrition plays in disease occurrence. And probably the most classic examples are really coming from coronary heart disease in general, where there are giant differences in the likelihood of a person developing disease across the US, despite the fact that we have a relatively homogeneous genetic background that can only be explained by lifestyle differences, of which one of the biggest factors obviously is the eating pattern. And when we look at Japanese patients and whether they develop CHD in Japan or in Hawaii or in San Francisco, we see giant differences that cannot be explained at all by genetics. So we said, okay, well, then the answer is simple. we need to somehow build into our model how the molecules that reach us through our eating pattern interact with our cellular networks, with our proteins, genes, and metabolites, and how they affect health. And as we looked into that, we were in for some very interesting surprises. And I like to use this example to explore that. This is actually a very much explored, biologically much explored pathway that talks about how meats, for example, affect health. And there are multiple ways the meat consumption can actually affect health. There's a cholesterol pathway and so on. But one of them that has been very well explored lately has to do with the chlorine and L-carnitine consumption. That if you actually consume milk, you are consuming choline and L-carnitine, which is then transferred by your gut bacteria into TMA. Then the liver takes the TMA and turns into TMAO. And there's quite a bit of epidemiological evidence to show that individuals who have elevated TMAO levels actually have a much higher chance of coronary heart disease. So this is indicating really that that through this pathway, through the consumption of the meat, one can actually elevate the chance of having a heart disease. And interestingly, in the Mediterranean diet, as you know, there is an abundance of garlic and wine and olive oil, which bring to the diet allicin and DMB, which in turn have the possibility of blocking the gut bacteria's ability to create TMA, and hence the TMAO production decreases. This is a well-studied pathway. And the only reason I actually kind of bring it up is for us to understand how much can me as a patient learn about the presence of these chemicals in my diet. So if I would really want to make sure that my diet is free of the chemicals that lead to TMAO, I would like to know whether any of these chemicals are really part of that, of my daily eating patterns. And when we started to look into the databases, we realized that of the 6 chemicals involved in this pathway, only one, choline, is being tracked by nutritional databases. The remaining 5, you would not be able to figure out that you are eating it just by kind of looking at the existing databases. And that was very unexpected, and that kind of led to the concept of the dark matter of nutrition. Yes, there is an exceptional effort out there that is many decades long to map out what are the nutritional components present in our food. And the chief proponent of that is USDA, that systematically tracks and reports the presence of about 150 nutrients in our food. These are chemicals that we need in our diet for us to survive. These are chemicals that have direct connection to certain diseases. For example, if you look into garlic, USDA would report 67 chemicals as being present in it. However, when we look more carefully, it turns out that there are more than 20,000 compounds that are known to be present in the food. And only in garlic, there is evidence for about more than 2,000 components as shown in this one. So at the end, what we realized is that really there is a dark matter of nutrition, that only less than 0.5% of the chemicals that are present in the food are currently tracked by nutritional databases, and 99.5% of the chemicals are really go undetected, untracked. Now you would say perhaps they are not detected and not tracked because they don't really matter for health. But then we did a massive study by connecting each of these chemicals to known health effects in the literature, and we realized that there are twice as many health effects connected to the dark matter of nutrition than how many could be connected to the nutrients. This is not to say that the 150 nutrients are not important. Those are key for our nutrition, and we must consume them in order to stay healthy, but many, many other chemicals are really contributing to the health and well-being of individuals. So we set out actually to map out the dark matter of nutrition. The first step was to really look at the existing databases of how much information is available. For example, I mentioned already USDA that has for about 9,700 foods, information about 150 chemicals, but there are also other databases and books that over the last decades have been actually systematically collecting information on that. But we also went up and did mass spec on basil and other components, and we realized that the mass spec was actually detecting many, many chemicals that are not listed in any of these databases. So one of the big projects currently in my lab is to really understand what are the chemicals in our food, which is the first step necessary for us to really understand how diet affects health. How are we going to do that? How are we going to complete this periodic table of food? Well, take, for example, apple, which has a list of known chemicals, and there are many other chemicals that we don't know if it's present or not. But we actually have genomic information about apple that could give us information about the enzymes producing certain chemicals, so we can guess that they should be there or not, allowing us to fill in some of the details from genetics. And we also have similar information for multiple other plants that are phylogenetically related to apple, whether they are far or whether they are close to it. And even non-plants like animals and so on. So at the end of the day, what our plan is to use genomics to fill in the missing details, but then to rely on machine learning and AI to really kind of identify chemicals that are missing from certain plants, but they should be there based on the fact that based on their genomics, as well as based on the fact that very related organisms have those chemicals. And eventually we plan to use, and we're actively using mass spec to validate our predictions to see how accurate we are. This is an ongoing journey. The end of this would be really to create this periodic table of food. And so it would tell us, for example, if you pick up certain plant like basil, how many and what kind of chemical compounds in there, what kind of flavor compounds are there, and as I will show in a minute or so, what disease connections is the basil connected to, and based on the chemical composition and so on. But the question is, how do we go from this information, from the chemical composition of the food to health? And that's where network medicine can really help us. On one end, we actually built what we call the knowledge graph that collects all the literature that is available out there and identifies all the known chemical and disease connections. And this is an ongoing project. We'll be soon actually publishing the outcome of that. But to show you really how this works, I'm going to focus for a minute on polyphenols. Polyphenols are known to be very many positive health effects, and are typically present in fruits and vegetables. And the question is, how can we use network medicine to understand what are the potential health effects of a particular chemical, like a particular polyphenol? And for that, let's start with green tea, right? Whose active ingredient is EGZG. And that polyphenol has a couple of known targets, which I'm indicating over here. And they hit a certain number of proteins within the protein interaction network, the one that I showed you at the beginning of the talk. But parallel to that, part of the network medicine journey, we are also curating in what neighborhoods different diseases reside. So disease A, disease B, and disease C. And what we have shown in network medicine, that if the targets overlap in a statistically significant manner with a particular disease, then that particular chemical likely has an effect, beneficial or negative effect on that disease. So in this case, we would expect that ECGC would affect protein disease A, proteins of disease A, but not B and C because those are far away. And you know, explicitly when we do that, we identify the targets within the network of ECGC. And in this case, I'm showing you type 2 diabetes proteins that happen to be in the vicinity of the targets. Hence, we would predict that ECGC has an impact, potential impact on diabetes. And with that, we can not only focus on diabetes, but we can actually rank all the diseases based on their potential relevance to ECGC, or the ECGCs, or the T's potential relevant to disease. And this is the ranking list that we obtained. And what you actually see is that all of the top diseases that we predicted using a network medicine framework are known to actually be related to tea consumption, but we also have new predictions that need to be tested experimentally. Now, we actually went further, and we can, from this framework, we can provide direct experimentally testable predictions. And in this case, the methodology predicted that rosmarinic acids, RA, was a candidate for vascular disease. And not only that it was a candidate for vascular disease, but it actually told us that if it affects coronary heart disease or vascular disease, it does so by affecting platelet function. And it also told us the precise pathways through which that happens. And so we ended up actually going and taking human cell lines and adding rosmarinic acid to that and measuring explicitly whether the perturbations within the cell lines happening in the way the model predicted. And the answer was yes, the experiments fully confirmed the model predictions. So where does this lead us? Well, our project is an all-encompassing project that goes from all the way to collecting all the existing information that link food chemicals to wellness and disease, to all the way to mine the literature for the chemical composition of food ingredients, aiming to complete this periodic table of food. But we also think a lot about food as drug and to identify food molecules with beneficial health effects. We explore food and drug interactions. And most important, this framework offers a way for us to identify these precise molecular pathways that link food chemicals to disease. So it's not just an association or a correlation that tells us where is the mechanism and gives us experimentally testable predictions. And lately, we also explore processed food a lot in the lab, aiming to understand how the nutritional and the chemical changes happening once the industry processes the raw ingredients. I would like to end by showing some of the papers that we published in the last few years. And the two that I'm showing in the red are focusing on the nutritional dark matter that I spoke at the beginning, as well as the polyphenol work that I kind of mentioned at the end. And this would have not been possible without a fabulous team that worked on this problem within my lab, as well as a fabulous group of collaborators like Joe Loscalzo from medical school, Darius Mozarafian from Tufts University, and Walter Wild from Harvard School of Public Health. Thank you very much for your attention.
I'd like to thank the NIH for the opportunity to participate in this meeting. I have no conflicts of interest. I'd like to start with a quote that I read in one of Karen Teff's papers. This is from a scientist who said, "It is not accidental that all phenomena of human life are dominated by the search for daily bread. Precise knowledge of what happens to food entering the organism must be the subject of ideal physiology, the physiology of the future." This was Ivan Pavlov's Nobel lecture in 1904. So now we're talking about the physiology of the future through precision nutrition. I'm going to spend the next couple minutes talking about what happens to food once we eat it and how to test this metabolism. But before I do, I thought I'd share with you one data set of many in the literature that kind of captures how Americans eat. This is a study by De Castro from 2004, in which he analyzed 7-day food records from 900 subjects. You can see from the analysis that carbohydrate intake is relatively steady throughout the day, although we eat more at the dinner hour. By contrast, Americans tend to eat low-fat breakfasts, and so fat rises dramatically at lunch. Protein intake has a similar pattern as carbohydrate intake. And this study was conducted at a university campus, so fortunately for us, De Castro also collected the alcohol data, and you can see here that it rises throughout the day. What's striking about these data is how we consume nutrients all at once, and therefore human physiology has to process all these macronutrients simultaneously. When we think about quantitating systems flux, we know that humans are systems analyzers. We watch the weather report on the news, and we are not challenged by the presentation of that complexity. There's lots of variability, lots of data, and the weather model helps us predict the future. Thus, our analysis of the complexity in the world should come naturally But somehow in biology, modeling of systems has not been universally accepted. Rather, the scientific culture has continued to focus on breaking down systems using the reductionist approach. Using this approach, nutritionists for many years have measured micronutrients and macronutrients in the fasting state, thinking that this was a steady state. But actually, it's non-equilibrium, as pointed out by physicist Richard Feynman. This is a system being in equilibrium when all the fast things have happened, but the slow things have not. And indeed, the longer one fasts, the more the body adapts to the lack of food intake, particularly the liver, which is making more and more glucose to maintain fasting glucose concentrations. Now, if we took 100 random people off the street and put them in a clinical research center and measured their blood glucose levels, you'd find that at that time, there was a high variability between them. But if we fasted them for 24 hours, gave them nothing to eat and only water to drink, and then measured blood glucose, that variability would collapse down to a very narrow range. Even if that group of people had some diabetics in it, no matter what age they are, no matter what ethnicity or what their body weight is. Thus, the premise of our lab is that the ability to appropriately become fasted is maintained in diseases like obesity and insulin resistance. By contrast, the metabolism that causes disease occurs at the transition from the fasting to the fed state. It is in eating that the body has to respond to these changes, and in health, this is done very quickly and efficiently. So diurnal metabolism is this alternating flux between fasting and feeding. You can see here that fat metabolism is high in the fasting state. And then when we consume amino acids and carbohydrate, their use goes up and fat use drops. Then right before lunch, we approach a fasted-like state, and then we're fed again. Thus, the diurnal metabolism is that during the daytime, we're awake and eating and moving around. And at nighttime, we're sleeping and metabolism slows. What's absolutely amazing to me is that we are completely unaware of these transitions. Here's one example. This is an appearance of ingested glucose versus the release of endogenous glucose. Down here in the lower left-hand corner is meal glucose turnover in the open circles, and you can see that once carbohydrate is consumed, their turnover increases and then drops slowly. This slanting line here of filled black circles is liver glucose production, and it takes almost 2 hours for the liver to reduce its glucose output. The result is the combination of these two fluxes and results in the peaks in blood glucose that we see. And these are dependent then on the restraint of the release of endogenous glucose while the meal glucose is entering the blood, and the storage and use of meal glucose. It's tests like this that are quite informative in terms of metabolic health. Now, one other fate of meal glucose is that it can go to the liver and be made into fat. And this is a process called de novo lipogenesis. I'd like to show you the variability in a number of subjects. These are all men, and we measured metabolism over a 24-hour period. Each line on this graph are data from a different subject. You can see that this subject in the upper left-hand corner had very high lipogenesis at midnight. And indeed, in every subject we've measured, we've found that this pathway peaks at midnight, between midnight and 1 a.m. Then, as carbohydrates are becoming limited in the body, lipogenesis begins to fall. And when we eat, it increases. And in some people, very quickly. Look at this subject with this pink line, denoted by this pink line of data. See how fast lipogenesis increases. We have looked at a number of characteristics that might predict the control of this pathway, and we found that age does not predict it, and neither does body weight. Lipogenesis is increased in insulin resistance, but much work needs to be done to understand how this pathway turns on at the onset of eating. So I've shown you a pathway where glucose is made into fat, and we eat meals where glucose and fat and amino acids through proteins are taken in at the same time. The interactions of those nutrients will be important to understand as we understand response to feeding. This is data from a paper by Robertson where they fed 2 oral fat tolerance tests. And they did this first by feeding people a high carbohydrate evening meal before the test the next day, and the second time by feeding a high fat evening meal. When the high carbohydrate evening meal was fed, you can see that fasting triglycerides are elevated, and the clearance of blood triglyceride from blood is reduced by pre-carbohydrate feeding. In this paper, they also fed 2 oral glucose tolerance tests to the same people and with the same high carbohydrate and high fat pre-meals. Again, you can see the high carbohydrate raises fasting triglycerides. So these ideas need to be taken into account when making tests for precision nutrition studies. What was interesting to me is that when a high fat evening meal was fed and an oral glucose was fed, we saw an increase in triglycerides in blood. Now, no fats had been fed in the morning, so I asked, where does that come from? And Robertson and others, including our group, have now determined that this fat coming into the blood in the morning is pre-stored from an intestinal fat pool that was derived from a previous meal. This phenomenon has been called the second meal effect, and indeed an electron micrograph of intestinal tissue shows fat droplets in the intestine 5 hours after a meal has been consumed. We've found that this fat can remain there for 16 hours overnight. A postdoc in our lab, Miriam Jacome-Sosa, put stable isotopes in lunch and dinner and was able to show the magnitude of the effect of lunch fat coming in right at the onset of dinner. And we've also shown that the release of this fat is taste sensitive, which suggests it's vaguely mediated. There's much work to do. Justine Musinski in our lab has recently analyzed data by adding the isotope of fat to a meal and analyzed LCMS data, mass spec data of triglyceride in blood. We performed this study doing a low fat, medium fat, and high fat tests. I'm just going to show you the high fat data. One can appreciate that the average line there in the black really doesn't define any one subject. And indeed, triglyceride absorption rates are the most variable of any macronutrient. They're very important to study, and this is because high plasma post-meal triglycerides are an independent risk factor for cardiovascular disease. They're a key feature of atherogenic dyslipidemia. And elevated triglycerides have the strongest association with cardiovascular disease when they're measured after meals. So now there are optimized methods for measuring dietary fat processing, and a number of investigators are ready to investigate gene by environment interactions. So I've shown you that the macronutrients influence each other's metabolism, and this needs to be kept in mind during study design development. But what about alcohol intake? Well, we've just completed and published the Tailgate study, Differing Metabolic Effects of a Bout of Excessive Eating and Drinking. In this study, subjects had liver fats measured by magnetic resonance spectroscopy in the fasting state, and then again after 5 hours of consuming Tailgate foods. We used a breathalyzer to keep their blood alcohol levels steady. They were inebriated during the study, and thus we kept them in the Clinical Research Center until the next morning when they had dried out, and then they could go home. We measured subjective intoxication on the y-axis with a 10 being more drunk than I've ever been and a 0 being not drunk at all. You can see they achieved a level about 4.5. Liver MRI was measured in the fasting state in the morning, and then that was compared to 5 hours of feeding. To my huge surprise, there was no increase in the mean level of liver fat. And I had hypothesized that every subject would get fatty liver with this many calories. So graduate student Majid Saeed plotted the data, and then we crudely separated the subjects into those whose liver fat increased and those whose liver fat decreased. Their caloric intake was not different. The individuals whose liver fat increased tended to consume actually less alcohol than those whose liver fat decreased. Their carbohydrate intake was also greater, although not statistically significant. We looked at many characteristics and hormones and metabolites, and the only thing that discerned which group these subjects were in were whether or not their liver made fat out of these substrates. And indeed, the greater the lipogenesis, the greater the accrual of liver fat during the study. So to summarize current concepts and knowledge gaps, with regard to alcohol, we know its intake is highly prevalent in the population, and there's a complex relationship between intake and health and disease. Indeed, small amounts of alcohol intake and moderate levels may reduce risk for cardiovascular disease. However, we know that larger levels of intake correlate with excess food intake, excess body weight, and metabolic dysfunction. This year, during COVID, average alcohol intake in the population has increased, and this is of concern. There are gaps in our understanding. How does alcohol affect the processing of other nutrients? What influence is there of physiologic and social factors that drive alcohol metabolism? We've now developed high-throughput isotopic methods for measuring metabolic flux. These methods are being scaled up to allow meal studies to be conducted by investigators to measure metabolic kinetics during precision nutrition. With regard to glucose, targeting glucose kinetics for disease has already been established. We've heard previously we need to determine how the timing of behaviors impact glucose kinetics. For protein, I didn't have time to talk about protein today, but sarcopenia or muscle loss is a highly active area of investigation. And finally, for lipids, major gains in our understanding of lipids absorption have been made. We need to understand how people vary in post-meal lipid processing and how does that impact their future disease risk. I want to thank you for attending this conference, and my appreciation goes to the research subjects who volunteered their time and all the brilliant mentors and trainees who've contributed to my learning. Thank you.
It is a great honor and privilege for me to speak at the Precision Nutrition Conference on Sensory Nutrition. I have nothing to disclose. I am a registered dietitian in an interdisciplinary department focused on health promotion sciences. Sensory nutrition is key to understanding inter-individual response in dietary interventions. Consumers consistently report that taste is the primary driver of what they eat. Sensory nutrition includes the interplay of receptor genetics, receptor density, fitness of nerves that carry the message to the brain, and perceptual integration that mediates food and beverage sensations, how much we like these sensations, what we choose to eat, and ultimately our health. This diagram illustrates the mechanisms involved in regulating feeding behavior via the gut-brain axis. Nasal oral sensations from foods interact with the environment to influence hedonic responses and appetite, while extraoral taste sensations interact in the gut to influence metabolic processes and hunger. I'm going to support that taste or sensory signals are a biomarker of health that work through mediating habitual dietary behaviors. There is large variability in sensory perception of foods and beverages, which ultimately influences our risk of diseases and conditions. I will recommend feasible and valid assessment of this variability for intervention studies. The chemical senses involve responding to chemicals via receptors and sensory cells. Oral shapes and particle sizes stimulate mechanoreceptors. If we were together, I would have you sample a gourmet jelly bean, first with the nose pinched and then unpinched, paying attention to the sweetness and flavor changes. Unplugging your nostrils gives you that aha experience of what no word in the English language represents. Flavor is beyond true taste, the perception of salty, sweet, sour, bitter, umami, to smelling through the mouth, the major component of food flavor. More complex foods present diversity of texture, temperature, chemesthesis, or chemical irritation. The sampling of food with nose plugged and unplugged shows the taste-smell confusion described by Rosen in 1982 and shown with functional MRI that flavor is a unique and integrated percept packaged in the orbital and insular cortexes and integrated with cognitive, emotional, motivational, and craving circuits, as well as gut signals. Because of this unique flavor package. Perceived sweetness of a coconut jelly bean, for example, is increased from nosed plugged to unplugged with the sweet coconut volatiles stimulating olfaction and processed as enhancing sweetness too. The sensory signal is highly variable normally and then with alterations. The variable signal should be measured in intervention studies, but isolated from metabolic testing because of potential cephalic phase responses. My talk is organized by these recommended measures. The first recommendation is the assessment of self-reported smell and taste perception following the N-Haynes protocol. In this protocol, participants were asked about smell and taste problems in the past year, loss with aging, and phantom sensations. 23% of adults 40 years and above reported smell alterations and 17% reported taste alterations. And an analysis by Lee and colleagues, those who reported smell alterations and taste alterations had less healthy diets by the Healthy Eating Index. From international collaborations and a publication in Press, perceived changes in smell was the top predictor of a positive COVID-19 diagnosis among individuals with respiratory problems. From the N-Haynes and comparison between measured and self-reported smell dysfunction in the 2x2 table, self-report underestimates the level of olfactory dysfunction. There are people who just do not realize they have olfactory dysfunction, likely because it's not measured routinely. Conversely, some individuals notice change in smell, but don't meet the criteria of olfactory dysfunction and are at greater risk of the least healthy diet behaviors. Thus, the second recommendation is to screen for smell dysfunction. The sense of smell is very vulnerable to damage with limited receptor fields that is very exposed and a single cranial nerve to carry the signal to the brain. There is great risk of olfactory dysfunction with aging, as well as with health and economic disparity. Olfactory dysfunction can result from blockage of odors reaching olfactory receptors, loss of sensory receptors, viral damage of the sensory neurons, as well as neurodegenerative disorders that damage the olfactory cortex, such as Alzheimer's disease. The COVID virus likely damages the sensory neurons, as well as unknown factors to result in smell dysfunction. From the N-Haynes and with an odor identification task, the prevalence of olfactory dysfunction is about 12% among adults 40 years and older and shows significant age-related increases. To reinforce, we smell through the mouth as the primary component of food flavor. Mouth and chewing motions release flavor volatiles and pump them to receptors behind the bridge of the nose. Eating too quickly, dry mouth, and oral health problems can diminish food flavor, even if the sense of smell is normal. Thus, we need to measure the sense of smell or screen for the sense of smell through the nostrils and through the mouth. There has been exciting new technology that has come out to improve the simplicity and quality of olfactory screening. One is this new lift and smell technique, using a single odor with detection, identification, and intensity tests. The other is to use the gourmet jelly beans with the nose plugged and unplugged. The third recommendation is to measure taste intensity with the N-Haynes protocol plus a probe of taste genetics. To capture variability in taste and oral sensation, evidence supports measuring phenotype as it reflects multiple genes, epigenetic, and regulators of oral sensation. The testing should involve multiple bitter and sodium chloride intensities. There is much scientific evidence that supports bitter intensity captures diversity and overall oral sensation, a variation in normal from individuals who live in a pastel food world to those who live in a more neon food world. 25 receptor genes mediate bitter taste and show large variability. Some bitters bind primarily to one receptor gene, such as propylthiouracil, PROP, to TAS2R38, and others, such as quinine, the bitter of tonic water, to multiple bitter receptors. Perceived intensity of concentrated taste vary across a healthy sample of individuals, ranging from weak to above very strong. The most variable are the bitters, especially propylthiouracil. Some individuals taste very little from concentrated PROP, whereas others experience intense bitterness. Bitter intensity has the ability to capture normal variation as well as dysfunction. Measuring phenotype captures more variability than genotype. especially to understand the perception of individuals who live in a more vibrant oral sensory world. There are bitter receptors throughout the body, including the gut. These bitter receptors are uncoupled to perceptual experiences, yet are involved in enteroendocrine hormone secretions and metabolic controls. Evidence supports that the variability in response to oral bitterness reflects the receptor variability throughout the body. The taste system is very redundant, with 3 cranial nerves carrying taste information to the brain. Total agousia is very rare. More common is variability in taste on the tongue tip, which was included in the N-Haynes protocol. Recommended for intervention study is a bitter and sodium chloride probe on the tongue tip and whole mouth, with an additional whole mouth probe bitterness probe. Tongue tip taste is altered with common exposures that damage taste from the chorda tympani nerve, intensify oral sensations from the back of the tongue and in the whole mouth to alter oral sensations and show associations with diet and weight outcomes in children and adults. There are challenges to measuring phenotype. As example, variation in taste can associate with body weight, but excessive adiposity also alters the taste system. The food environment influences the expression of taste genetic effects. As shown in Keller's lab with preschoolers, unhealthy food environments associated with a blunted effect of PROP on liking for vegetables, yet show that bitter insensitive preschoolers have the highest risk of elevated BMI Z-scores. Food characteristic also blunt taste genetic effects. In plain broccoli, you see less broccoli consumption in the bitter sensitive preschoolers, but with dip available, the bitter sensitive children eat broccoli to the same level as the bitter insensitive preschoolers. The next recommendation is to use standardized and rigorous measures of intensity following the N-Haynes protocol. The goal is valid measurement of perception and maximizing the ability to detect inter-individual differences. The labels on the scale do not have inherent meaning and are elastic based on the modifier or directions. From the father of modern psychophysics, mice may be called large or small, and so may elephants, and it's quite understandable when someone says it was a large mouse that ran up the trunk of the small elephant. We can apply this to detecting inter-individual differences in oral sensation. Without standardized instructions or context of the top of the scale, those bitter insensitive could appear to perceive greater intensity to a taste such as table salt than those bitter sensitive. In reality, with generalizing the scale top to all sensations, the bitter sensitive individuals are correctly shown to perceive the highest sodium chloride sensations. N-Haynes includes standardized script to instruct individuals on how to use the general labeled magnitude scale, practice rating with non-oral standards, and it includes individuals only who could appropriately rank perceived or remembered non-oral sensory standards. The final recommendation is to survey assess food and beverage likes and dislikes. When consumers report that taste drives what they consume, it is really that they like the taste and that liking drives consumption. As an example of this evidence, less intensity from bitter taste markers associates with greater fat-sweet preference, which is the strongest predictor of central adiposity in a sample of women from a lab-based study. Here is a listing of reasons why to ask food and beverage liking to understand sensory nutrition impact on precision nutrition. Survey liking takes minutes to complete, which can encourage participation in dietary studies across diverse groups of individuals. As one reason why to use a liking survey liking shows strong correlation with sampled food preference. Difference in food preference related to taste receptor genetics, as shown with variability in bitter taste perception of grapefruit juice, can be detected by sampling the grapefruit juice, but also with survey preference, albeit with less precision. We need to apply the same psychophysical principles to testing food liking. For example, if we only survey liking for vegetables, and then survey liking for sweets, we might get the wrong result that a preference or a preferred vegetable, such as tomatoes, are liked more than a less preferred sweet of butterscotch pudding. Needed is to generalize the top of the liking scale with directions and have participants rate foods as well as non-foods in the liking survey. In multivariate modeling, a latent bitter variable of quinine on the whole mouth, tongue tip, and probe explained variation in central adiposity that was partially mediated by a liking-based diet quality index, and this liking-based diet quality index fully mediated the relation of taste to HDL cholesterol. We have advanced liking-based diet quality indexes that are conceptually based on food groups and empirically weighted to increase the explained variance in cardiometabolic risk factors in young adults. The precision of dietary assessment is increased by identifying discordance between liking and intake as dietary restriction or trying to improve the diet by eating less preferred healthy foods. In summary, there's much opportunity to leverage sensory nutrition for understanding inter-individual differences in responses to dietary interventions. I have suggested feasible and valid measures of variability in sensory nutrition. I further recommend collaboration with scientists in sensory nutrition and to refer to this new publication that reports on an NIH workshop of sensory nutrition and disease. This publication expands recommendations for food-based probes, recommends interdisciplinary collaborations to understand the effect of extraoral reception and gut-brain axis responses, and to expand clinical and population-based research. Thanks so much.
Okay, thank you for those great talks. As we assemble here online, Dr. Duffy, I'd like to send a question to you first, if you're available. There are a couple questions in the chat. Are you ready? Dr. Duffy.
I believe so.
The question on the chat is for Dr. Duffy. Earlier today, we heard that there was a circadian variability to hunger sensations. Is there circadian variability to taste or to specific tastes? Do we know this? If so, how do we account for this in our assessments? That's a pretty hard question.
That's a very interesting question, and thank you so much for the question. I am not sure about a circadian rhythm to taste. I know that the taste in the gut, they're nutrient sensing. So I wouldn't be surprised if there wasn't some kind of rhythm of up and down regulation. In our studies, we always ask perceived hunger, not just how long somebody has gone without eating, but perception of hunger, which we always covary in any analysis. So at this point, I'm not certain that there are definitive data, although I probably am wrong, but I would always ask perceived hunger before any type of metabolic testing, just to control for that.
Excellent. And can I stay with you? One of our questioners is asking if there are strategies to increase flavor perception in the context of olfactory dysfunction. Can we lift weights to improve our sensory function?
That's another excellent question, and the response is that there has been previous research on trying to enhance flavor perception by enhancing the volatiles. But if you don't have a good sense of smell, it's not certain if that helps. Just remember that smell and taste and touch sensation and irritation sensation is processed uniquely in the brain. So even small amounts of sweetness can enhance overall flavor and vice versa. Small amounts of flavor can enhance perception of sweetness. And so the the response is it's probably not uniform, the person's response to enhanced flavor perception, but if it does help, that is important. It's also, and I'll just say one more thing, that our processed foods aren't necessarily so flavorful. There's a lot of salt and sugar added as opposed to if you cook at home and you work to enhance the flavor of food. So we eat with our cooking, we eat with our eyes, we eat with our crunching or texture, as well as the overall flavor sensation.
Yeah, thank you very much. The next question is for Dr. Barabási. In your research, the person is asking, are you also considering the variability in the chemical compositions of foods due to soils and environmental characteristics? Did you hear that question? Dr. Barabási, did you hear that question?
I had a little internet connection problem. I'm calling in from Hungary.
Oh, thank
Sorry.
you. I can repeat it. The question is, in your research, are you considering the variability in the chemical composition of foods, for example, from the soils and environment? We'll see if that you can hear it. That.
I'm assuming you can hear me now, right?
Yes, we can. Yeah.
I'm assuming you can hear me now, right?
Yeah.
So we would love to do that. And actually, we had a collaboration and a dream to go in that direction. And to actually work with people who are growing some of the plants and consider the variability in the growing conditions and see how it affects the nutritional content and the chemical composition of the food. That being said, we're far from that. I think we'd be right now happy and delighted if we could get as accurate description of the food composition of the main foods that we eat, kind of the generic versions of it. But once we get over that, we would probably want to do kind of mass spec on kind of plants grown in different conditions and see how those effects that you mentioned affect the composition.
Thank you. I'm going to respond to a question that's been addressed to me. Someone is pointing out that alcohol intake, that those calories displace fat oxidation. So their question is, where does the fat go if not the liver? And we and others have shown that quite a bit of meal fat is cleared both to the muscle and also, of course, to the adipose tissue. And so that what correlated with fatty liver in our case was not dietary fat clearance, we don't believe at all, that it was alcohol transmission into fat in the liver. I'd like to go back to Dr. Barabási, if you can hear me. You had said that 99% of constituents in food are dark matter, but the known nutrients in current databases are 99.5% of the weight of the food consumed. So we still know very little about the physiologic effects of minor chemicals. Can you comment on this?
Oh, this is absolutely true. And that's one of the reasons we actually have these nutritional databases, because they capture the selection of the 100 or 150 nutrients that are captured by USDA and other sources are really what give the bulk of the weight, as well as the nutritional components and the energy sources that we need to kind of survive when we eat food. That being said, it's not only quantity that matters, right? A very small amount of nutrients can actually, or chemicals can affect our health. One of the things that was actually surprising to me when I started working on this is the realization that when it comes to the nutrient composition, that spans 9 orders of magnitude in 100 gram food, where we're going to like 10 to the minus 7 grams per 100 gram actually when it comes to vitamin E, all the way to, you know, almost 100 gram when it comes to water. And many of the vitamins role in our health come in such a minute quantities that they really don't contribute to the bulk. So in a way, it's not about the weight. It's not about really how much of it is there, but whether we actually those chemicals had health effects, and very, very tiny amounts can make a big difference. And that's one of the reasons actually why so many chemicals with known health effects are outside of the nutritional kind of canon. What our analysis has shown, as I briefly mentioned, that there are twice as many health effects associated with chemicals that are outside of the nutritional canon, that is, they're in the nutritional dark matter, than they are actually within the 150 chemicals currently mapped out by databases. So the goal is to really go in that direction and understand, no matter how little it is, whether it has health effects, and if it does, why and how.
Thank you very much for that. A related question is that foods tested in research studies might emanate from different sources. For instance, one basil added to a meal might be different than another. And so one of the ways we account for this is by banking, freezing ingredients, using standardized menus, that kind of thing. But that's more difficult over a longer period of time. And so it seems that the heterogeneity of ingredients also needs to be taken into account, and that seems to be a pretty heavy lift.
It is, but it is not in a way. I mean, let's be clear. First of all, it's the genetic makeup of each plant that determines what is there or not, and that's the first question: is whether a chemical can be produced by the plant or not, and that's typically provided by the genetics, or at least pre-encoded. Not easy to get it out, but it's in there. Then comes the next question: is that if the organism can produce a certain chemical, and that does accumulate, because sometimes they are just transients and being produced and consumed right away by some other biochemical reactions, and then really the question comes, how much of this is being accumulated? And there again, we have a result, hopefully we will release it very soon, to show that when you look at these 10 to the 9 gram range of chemicals, for any chemical that is in the food, the variability is relatively limited, which is surprising, but it's true. That is that when it comes to vitamin K, you don't find 3 orders of magnitude differences from one food to the other. Everything that contains vitamin K or vitamin E will actually roughly contain a comparable amount, and the variability is a little bit predictable of how much variability could actually be there. So in a way or the other, the quantification is a major problem, but the variability is not so major. Now, there is actually variability associated with what we consume. So certain chemicals can be produced by the plant, but may not be in the part of the plant, whether that's a root or a fruit that we consume, and one has to consider that as well. But I actually believe that these are all treatable questions. So if we're willing to kind of get out of the comfort zone, which is kind of looking at the nutritional component, we can develop a whole set of tools to address these questions in a systematic manner.
Yeah, thank you. I'd like to finish the speaker's discussion with Dr. Duffy. You talked about taste in children, and I wanted to know if you could kind of highlight other areas in the lifespan where this is really important in nutrition research.
Absolutely, and I just want to say that studying sensory nutrition is fascinating and is highly, highly interdisciplinary. So I'll just try to give a little just overview, but in general, the senses of taste and smell are formed well before birth, and we're born liking taste, the sweetness of sugar, and disliking bitterness. But we actually learn to like things in utero, and there's been some fascinating work about that. And then we learn to like things developmentally early in life. And smell is we learn to like things coupled with first calories and then repeated exposure. And then throughout life, the sense of smell is much more vulnerable to damage than taste, because taste is very redundant in its system, but the sense of smell is vulnerable, and we know this with all of this information about smell loss and then taste, but it's really the flavor of food lost with COVID because of the vulnerability in the smell system. And then probably across the lifespan, taste is upregulated during childbearing age to support a healthy pregnancy, and that fitness may decline perimenopausal. And so older adults likely can have a loss of their sense of smell, although it's not absolute. If you're healthy and you keep up good physical activity and you have access to good health care and living environments. But the sense of taste can be altered with damage to the tongue tip taste, and it can alter sensations and give phantom sensations. So that's kind of a life course event, but if there's more interest in that, it's just a fascinating field to be in, and lots of wonderful scientists in sensory nutrition.
Thank you very much. We're going to turn now to our panelists, and we're going to start with Dr. Saroja Voruganti, who has some slides to share with us while she speaks. Over to you, Saroja.
Thank you, Elizabeth. I'm really thankful to the NIH and you for giving me this chance. The points I wanted to talk, actually, they have been talked before, and Dr. Ottawa's talk, we are complicated, and I want to stress a little bit on that. So we know that people differ in their requirements, and either for responses to nutrients or for their requirements, dietary requirements. And then we also know that one size doesn't fit all. And the heterogeneity in this metabolism, nutrient metabolism, is contributed by variations in genetics, epigenetics, and microbiome, lifestyle, dietary intake, food behavior, circadian rhythms, and environmental exposure. We have seen that. And so, but we have already seen these are all the factors which affect the responses to nutrients. So it is very important to stratify people into different subgroups. Individualized nutrition, to go to an individual may take some time, but stratifying people on responses versus non-responders makes a lot of difference. I'll show you one example, I'm basically doing more genetics and epigenetics. So we see a lot of difference between even within responders and responders versus non-responders. So I'm at UNC Nutrition Research Institute, and where the faculty is focused on identifying various bases like genetics, epigenetics, metabolomics, microbiome bases for interindividual variability in response to diet. My own research is focused more on genetics and epigenetic factors, looking at nutrients and combination of nutrients, how they affect purine metabolism and related disease risk. So I want to show you one example why compiling all responses together shows a lot of noise. And can you change the slide, please? So this is where I chose this example because interindividuality to serum urate in response to fructose intake has been shown by many studies, and it's nothing new which I'm showing. But if you see that carefully, you see that there is a 40% of people who show no response at all. They just don't go up at all, which has been shown that in response to fructose, uric acid goes up. But in this case, it just doesn't show up. We could not do much more on the non-responders because the sample size was too small. But when we look at the responders, you'll see that then again there is so much variability. Next slide, please. And in that itself, when you start looking at SNP-wise, this was a SNP which was linked to the variability in responses, and you'll see that having a CC or TC or TT, they respond differently. Having 2 copies of T, actually the uric acid lowers much, comes back to the normal level or goes down the level after 3 hours. So this is a very simple example where I want to show that there is so much variability, and like Dr. Ottawa said, we are very complicated and there's so much, but stratifying responders and non-responders is a very good first step in the direction of precision nutrition. So, and then the other point I wanted to raise that I do a lot of genetic studies, and I see that we do, now we started recognizing the importance of diverse populations. We do include several ethnic populations, but still we don't look deeper into them. Like South Asians are all grouped into one, Latinos are all grouped into one, but there is so much genetic heterogeneity in each population. So basically, next slide please. To summarize, I just want to showcase on 3 points. Dietary intake, that we need to do better measures of dietary intake. I don't want to stress on that because a lot of slide presentations have stressed on that before. But the second point is integration of omics data and groups, either responders or non-responders or various ethnic groups, is very important to going towards the precision nutrition. And finally, I think communicating proper information to the end user is very important to get measurable health benefits. We should not be giving them conflicting results or conflicting information. So we need to put together everything and come up with a reasonable step which the end user or a person can follow the steps how to do towards the precision nutrition. That's all I want to say.
Oh, great. Well, thank you so much for having me here today. My name is Krista Varady. I'm a professor of nutrition at the University of Illinois, Chicago. And I've been studying basically intermittent fasting for the past 15 years or so. And I run pretty much all my studies in human subjects. And we're really trying to look at how these diets can help people lose weight and then manage metabolic disease risks. So help, lower diabetes and heart disease risk factors. And I actually, to kind of tie this back into precision nutrition, since it's not, my exact field, I became interested in running intermittent fasting studies because I started with a general interest in different weight loss diets. And then, of course, one of the first things we were studying was calorie restriction, so kind of more like traditional dieting. And what I started noticing was that, sure, people could lose a bit of weight with daily calorie restriction. So that's when you cut out 500 calories or so per day. But then I noticed that a lot of people just started failing or dropping out of these studies or just quitting these diets after a month or two. And I kind of started talking to people to figure out what was going on. And a lot of people just got really sick of vigilantly monitoring calories day in and day out. With those diets, you're given a calorie goal, if any of you have tried it, of 1,500 calories or what have you. And then you kind of spend all those calories and then spend the rest of your day just being miserable because you can't eat anything else. So I thought to myself, well, do people really have to diet every day to lose weight? Or can they potentially have a day off of dieting and then that would be alternated with a day of kind of more heavy restriction? Next slide, please. So that's what alternate day fasting is. So alternate day fasting is a form of intermittent fasting. There's a bunch of different types. There's time restricted feeding, which I won't go into, but alternate day fasting is kind of one of the major types, and it involves a day where people eat about 500 to 600 calories. That's the fast day. You can kind of place that anywhere you want in the day. And then that's alternated with a day of just eating freely. And so we thought, my lab thought, well, maybe this will help people stick to these diets for longer because you get that free day every other day where you get to feel normal. You don't have to constantly put stuff in an app or food record or whatever. And so we ran our first study, which we started actually in 2010, but it was just published a few years ago. Next slide, please. And the point of the study was to really see, well, does alternate day fasting or do these intermittent fasting approaches help people to lose more weight than a traditional diet or a daily calorie restriction diet? And unfortunately, we were completely wrong. They didn't help people. People weren't able to adhere to them better. We kind of had the same adherence. People are really good at sticking to dietary weight loss regimens for about 3 months. You can see in the first 3 months here. So this is weight loss over the course of a year. Both diets ended up producing similar weight loss, so about 5% after 12 months. So yeah, we were wrong, and that was unfortunate. But from then, when we kind of found this out, next slide, please. We then wanted to see, well, who exactly does well with these diets? So this is just kind of a plot of the individual people in the alternate day fasting group. So it's about 35 people or 34 people or so. And this is how much weight they lost. And we just kind of organized them by degree of weight loss. And interestingly, we found that 20% of people just did not lose any weight. In fact, they actually gained weight. Then we found about 40% of people, they kind of lost a little bit of weight, but it didn't meet the criteria for clinical significance. So clinically significant weight loss is about 5% or so. And then we found that the next 40% of our group actually did lose a significant amount of weight, so 5 to roughly 15%. And then next slide. And then interestingly, we found almost the identical pattern in that calorie restriction group. So again, around the same amount of people, similar inclusion and exclusion criteria. So we're dealing with kind of similar people. But of these 35 people, we saw almost the exact same pattern: 20% gained weight, 40% loss within that 1 to 5% range, and then another 40% loss over 5%. So then next slide, please. This kind of got me wondering, again, since my main thing is looking at intermittent fasting, one of the things that we've been looking at more recently is, well, who do these diets work for? And in general, how can we prescribe different weight loss diets to different people? We've looked at a couple different determinants of weight loss success for fasting and found some differences. So we found that postmenopausal women actually lose considerably more weight with fasting than premenopausal women do. We're not quite sure why this is, but maybe because they know what diets work for them, because they've had more time to play around with different things, different diets. And then maybe they just have more time on their hands because they may not have young children at home. So we're not quite sure why it works so well in that group in postmenopausal women compared to premenopausal women. We've also looked at other things related to kind of precision nutrition. So does it work better in men and women? We haven't found any differences there. However, I should point out that with a lot of our studies, it's about 80% women who join the study. So we're usually kind of underpowered to see that effect in men or any differences between men and women. We've also looked at eating behavior and appetite to see, maybe certain people respond better to fasting because they just have different appetite control. And so most recently, we discovered in that group that lost the 5 to 15% of weight with alternate day fasting after a year, that those people actually had lower hunger on the fast day, as well as kind of higher fullness. And then that was related to better adherence with that kind of 500 to 600 calorie goal on the fast day. And because they were more adherent, not surprisingly, maybe, they lost more weight. So maybe that's one of the reasons. But I guess, there's so many questions in this area. The main thing I'm really wondering is why are certain diets? Why do certain diets work for people? As you know, there's so many different diets out there right now. Anyone working in actual kind of weight loss clinical trials, like low carb is really popular, keto, paleo, Mediterranean diets. Intermittent fasting has become popular over the past few years for the general public. But it's interesting because clinicians, there's just these diets out there. Clinicians, how do we actually tell people what to do? It's, which one, it seems like what we do now is just let people choose what they want, and then they either fail or succeed and then move on to the next diet. But I think this field, particularly the weight loss dietary intervention field, where we need to go is coming up with what diets work for certain people. possibly based on like sex, menopausal status, glucose homeostasis, eating behaviors, potentially the microbiome. So we could like plug it into an app or something. And I know this is what the precision nutrition field is trying to do, but it'd be great, you know, if we could kind of focus it in terms of weight loss, because obviously obesity is still a very major problem. So yeah, you know, just to throw those questions out there for the panel discussion later on. Thank you.
Hi, everyone. My name is Erin Hanlon, and I am a research associate professor at the University of Chicago. And I wanted to touch on and expand on a few things that Dr. Scheer said earlier today, and that includes the effect of sleep deficiency on increased eating. So Dr. Scheer talked a bit about how sleep loss or sleep restriction can impact food intake. And I wanted to address that specifically. So we know from epidemiological studies that show that there is an association between short sleep and higher BMI. And we know from well-controlled laboratory studies that in healthy, young, lean individuals, ratings of hunger and appetite are increased. Next slide, please. Ratings are increased, as is actual food intake. Next slide, please. Sorry, it has gone forward. We know that ratings of hunger and appetite have actually increased when we study lean, healthy, young volunteers, and actual food intake is increased in these individuals. So sleep loss or sleep deficiency actually leads to an increase in food intake. And the question then becomes, what is facilitating this increase in food intake in these young, healthy, lean volunteers when we study them in the laboratory, when we know, as Dr. Scheer said earlier, that the energy cost of extended wakefulness is actually quite modest. And so some of the mechanisms that are contributing to this perceived increase in hunger and appetite and actual increase in food intake include changes in circulating leptin and ghrelin. Here we go. So leptin and ghrelin, both when measured in the periphery, when individuals experience sleep loss, move in the direction to promote food intake. Both of them do. Next. And we know also that the ratio of the increase of the ghrelin to leptin ratio after a period of sleep loss is highly correlated with increases in ratings of hunger. So one of the questions becomes, what individuals, because these studies were done in very homogeneous groups of individuals, they're young, they're lean, they're healthy volunteers, but what contributes to the changes in ghrelin and leptin that are observed in these individuals? Because we can see the higher the change in ghrelin and leptin facilitates a higher change in, or a higher increase in hunger. So that is one of our gaps in knowledge as it pertains to sleep loss's effects on food intake. Next slide, please. And I specifically study a particular endocannabinoid called 2-AG. And endocannabinoids are lipids that are synthesized within your body that act on receptors that can be found both in the periphery and the brain, and they're involved in energy metabolism, including adipose tissue, the endocrine pancreas, muscle, and liver. And you are most likely familiar with an exogenous cannabinoid, marijuana, that also binds to cannabinoid receptors. But we make endogenous cannabinoids similar to THC that also bind cannabinoid receptors. And in regards to endocannabinoids, much attention has been paid in recent years to the ability of the endocannabinoid system to control feeding and appetite and energy homeostasis. And some studies have shown that exogenous cannabinoids, for instance, like THC, can induce hyperphagia and a preference for highly palatable foods. And we know that the endocannabinoid system can modulate brain reward pathways that affect eating behavior via reward circuitry. And one of the things that we also know about sleep loss's effects on food intake is that it's specific to snack foods. People seem to report feeling hungrier and having a stronger appetite for carbs and fat and snack foods. And so that is what led us to look at the endocannabinoid system due to its effects on reward eating or hedonically driven food intake. And we also know that sleep restriction, as you can see here, leads to an increase in circulating endocannabinoid levels. And whether or not there is individual differences in endocannabinoid levels within a person is something that we still need to understand. Next slide, please. Because in that particular study where individuals were studied over a 24-hour period, either with normal sleep or with short sleep, those two studies were done about a month apart. But you can see here that regardless of the month long between the two sessions, that individual differences of a mean level of endocannabinoids across the subjects is very different. So you can see that even though it's the same within a subject, it's very different across the spectrum of individuals. And again, this was a very homogeneous group. So what makes one individual have a low circulating endocannabinoid level, whereas another individual has a higher mean serum concentration of endocannabinoids, is something that we don't yet know. And we also don't actually know where these endocannabinoids are coming from. We know that we can measure them in circulation, but all of those peripheral organs that I mentioned earlier that are involved in energy metabolism, like adipose tissue and the pancreas and the muscle and liver, all of those tissues have enzymatic machinery to create and degrade endocannabinoids. So what tissues are creating this, these circulating levels, and what contributes to this variation between individuals, which then could impact hedonic eating or reward eating in a normal state, but further in a state of sleep loss. So that's some of the questions that I have that I thought that we could discuss in the panel this afternoon. Thank you.
Okay, thank you for the opportunity to be a part of this stimulating work group on precision nutrition today. I'm Susan Malone, and importantly, I'm your final panelist for today. And I'm an assistant professor in the Rory Meyers College of Nursing at New York University, and I'm a registered nurse. So as a nurse, I was well trained in the 5 rights of medication administration. And briefly, that's giving the right medicine in the right dose via the right route to the right person at the right time. But in my earlier clinical days as a nurse, I really didn't think a whole lot about these 5 rights in regards to lifestyle behaviors like eating or physical activity or sleep. However, our research team is interested in increasing the precision of behavioral recommendations, and timing might be one way to do that. A research area that I'd like to open up for further consideration is how the timing and rhythmicity of lifestyle behaviors can be aligned with endogenous and environmental factors such as light exposure to promote cardiometabolic health in people's everyday lives. We heard several presenters speak about time in terms of the benefits of eating earlier versus later for metabolic health. Earlier today, we also heard about how cardiac events might be more prevalent in the early morning, as well as during the darker winter months. So when we think about potential underlying reasons for why some people respond to some behavioral interventions and others don't, I wonder about the dimension of time and the nuances of time, not only for the interventions, but also for the assessments that we do. And in thinking about that, what might the quote unquote right time be? Should we be thinking thinking about social time dictated by the clock? For example, should all participants in a research study have an oral glucose tolerance test at a certain time of day? Or should we be thinking about biological time dictated by our internal body clocks? And if this is the case, what might be the best way to determine what those internal body clock times are? An important consideration might be how the biological night, and by that I mean, the difference between melatonin onset and melatonin offset, but that may be different for morning larks, people who prefer morning activities, than evening owls, those who prefer nighttime activities. And if this is the case, should assessments like the timing of oral glucose tolerance tests depend on maybe something like the number of hours from a participant's habitual wake time? Or should we be thinking about solar time, dictated by sunrise and sunset? And if this is the case, how do we account for regional and seasonal variability? In thinking more about differences between people in terms of when they prefer to do things, including eating, I think of a quote that's been attributed to FDR, and it goes like this. He said, "I think we consider too much the luck of the early bird and not enough the bad luck of the early worm." And a research area of interest to me that could relate to precision nutrition is whether an eating at breakfast or even eating larger meals earlier in the day is as beneficial to an evening person as maybe it is to a morning person. Or might it even be that an evening person could actually benefit from skipping breakfast? On the other hand, there's a lot of data to suggest that evening types engage in more cardiometabolic risk behaviors like poor diets and physical inactivity. So should we be thinking about shifting earlier evening chronotypes earlier? So time-restricted eating is another area related to chrononutrition. But do we know what the ideal time window might be between the first and the last eating episode? Should it be 8 hours, 10 hours, 12 hours? And if solar time matters, would the durations of fasting perhaps be longer in the winter months? In closing, how do we know what a, quote, right time might be, not only to guide chrononutrition, but also other lifestyle behaviors? And could that right time be found in all of those clocks, social, biological, and solar?
Thank you, Dr. Malone, and all the panelists. I'm going to start with a question that was addressed to Dr. Duffy, but also came up in one of our last papers, and I would ask the others on the panel to comment as well if you have ideas about this. The question is about when you deny yourself, when a subject denies themselves a certain flavor or taste, do they increase their cravings for that taste? And we found exactly the opposite in our last study. Reduced carbohydrate cravings. Dr. Duffy, is that consistent with the literature?
That's an excellent question. So let me give an example, and this is work done by Rick Matties many years ago, that if you wanted to reduce your sodium intake, that it would take weeks to do it. And it wouldn't be that you tasted salt any differently, but that your liking for the salt would decrease. Same as, for example, after bariatric surgery. The major findings are that bariatric surgery changes your liking for high-fat, sweet foods. It doesn't change your ability to perceive them, but your hedonic value. And so the question also is very interesting, which I believe is yet to be found, is let's say you stop yourself from tasting as much salt. Is that the requirement to lower your sodium or your salt liking? And that's not really clear because, you know, some foods are, through wonderful techniques, taste salty but really aren't high in sodium per se, where other foods like tomato juice, for example, just don't taste that salty, and they have so much salt so that you're even in the middle of the night, you know, dreaming of drinking water because you're so dehydrated. So, you know, your body can respond, but it really is a shift in how much you like the taste or the sensation.
Krista, have you found or noted changes in taste in alternate day fasting?
We haven't measured changes in taste. We've just looked at changes in appetite. But I think we're definitely going to include that food liking now. It's a good idea. That scale.
I'm willing to share anything. You know, that's what happens when you have white hair. You can do that.
And then there's a general question for everyone. Has there been indications that are correlated to excessive sleep and BMI or adiposity?
I can start. So I know that there are some studies showing sleep extension has effects on appetite. So just like sleep restriction will increase appetite, there are some studies, again, in healthy, lean volunteers that show that sleep extension in individuals that typically sleep, I think, about 6 or 6.5 hours, it'll decrease appetite. There is a little bit of discordance in the long sleep literature because very frequently excessive sleep is not just due to sleep need, but there's probably also another underlying cause of excessive sleep. So sometimes that can be difficult to dissociate excessive sleep when it has another underlying cause that isn't just long sleep to other things.
Yeah. Is there a correlation between excessive sleep and BMI?
Mm-hmm.
I don't believe that there is. I think it's the other one.
I'm not a sleep expert, but I believe I've seen that. We run time restricted feeding studies as well, so I think it's like more of a U-shaped curve with that. Like I think 7 to 9 is. I'm not a sleep. I'll hand it over to the sleep experts, but I think it's below 7 and above 9 is not great.
Yeah. Dr. Barabási, can you hear me across the sea here?
Yes,
there's a question for you. Did the data you presented suggest that garlic can counteract the negative health effects of eating red meat?
So that is not actually our data. That is a kind of a well-established result in the literature, and we do not have explicit data. Much of our work really focuses on bringing big data approaches combined with math-like methodologies to really map out what's in the food. And we are relying pretty much on your community, the nutrition community, to kind of get the traditional nutritional data. So that example that I use is really coming from the literature.
Okay, thank you. To all of you, how does socioeconomic status or environment influence your studies? Why don't we, so let's start with Dr. Hanlon.
Well, I actually am on the south side of Chicago where demographically speaking, we typically have a larger population of African Americans that we are able to study. And so in our particular studies, we run the gamut in individuals that we study. But it depends, again, on the study. So in our experiments, when we are trying to pinpoint short sleep specifically, and those studies will typically recruit young, healthy, lean volunteers. But in other studies that I'm involved in, we are able to recruit from the community as a whole, and we're not looking specifically for healthy young volunteers. And in that way, we are able to include all socioeconomic and racial demographics that are available to us. And like I said, very frequently, that is a large African American population for our studies.
Yeah, thank you. Saroja, how are you accounting at all, if you are, for socioeconomic status or environment in your studies of nutrition genetics?
Mostly in the analysis, basically, we collect the information on our epidemiological studies on access to food, and we've had I'm not an expert in this, but we have been collecting information on socio-demographics, and to be honest, I don't know much about it. I'm more of a genetics and molecular nutrition person, so but we have been collecting, and sometimes it can be biased because the studies we do, some people get recruited because of near Christmas, they need the money, so, or something like that. So, but beyond that, sorry, I can't comment on it.
Okay, Dr. Malone.
Yes, I can just comment a little bit. We have two sleep extension studies that we're recruiting short sleepers who have some of these chronic health conditions that comprise the metabolic syndrome. And again, we're still in the process of collecting data, but all these individuals live in the greater New York City area. And living arrangements are tremendous. It has a tremendous impact on their ability to get sleep of adequate duration, as well as unfragmented sleep, not only in terms of the number of people living in the household, but also in terms of the outside environment, their ability about whether or not they can even control the heat in the apartments that they live in. Sometimes the heat is so great, they just can't sleep at night because it's too hot. So these larger social determinants of health have a huge impact on one's ability to get adequate, high quality sleep. And I think an important area to think about with interventions moving forward.
Yeah, there was a question about how physicians can assess food intake or the amount of food. Dr. Malone, can you comment on, where we're going with precision nutrition? It's a long way before we get to the clinic, isn't it?
It is. I'm actually not sure I'm going to answer your question exactly, but I'm just thinking after today of all the things I could begin incorporating to get a better sense of this for my own research that's related to sleep and these health conditions. I wish there was a way that we could somehow move away from, the subjective dietary assessments that we're doing. And yet we also talk about that in the sleep world, and we think that actigraphy is the be-all and the end-all. And yet self-reports are important. They do matter, right? What somebody thinks they eat, how somebody feels they slept. So I don't think we want to knock those subjective assessments out entirely, but think about how we can include them and yet also perhaps get some more objective ways to measure food intake. So for me, with the interest in timing, I am very interested in perhaps using the actigraphy. They have the way you can tell when somebody eats with the wrist movement, and maybe that can complement what we're collecting using the ASA-24.
Yeah, there is a question in the chat about weight and sleep changes associated with psychiatric treatment. And one of the limitations of our work is that we sometimes have to exclude subjects on some medications because they affect body weight. And I think this is really tough. You want to start, Valerie, maybe? Some of these treatments affect taste, right?
Can you repeat again? And I apologize.
Yeah, I'm going to move on. We'll go to Erin, and if you can pick up her answer. Erin, psychiatric treatments and research, nutrition research.
Right. I was going to say, when you said that it affects eating, that we frequently exclude for psychiatric drugs because they affect sleep. So I don't actually know firsthand the effects. I know that some psychiatric treatments will abolish REM sleep, and so that's one reason that we exclude them in our particular studies. So I'm not terribly familiar with then their effects on the interaction between sleep and food intake, simply because we have to exclude for that very reason.
Yeah. Anyone else want to offer a comment here? Treatments for depression, for instance, can change body weight and metabolism. And we have a large portion of the population on these medications, and we're really missing out.
Can I add something? This is Valerie again. I apologize. The phone had rung in the background. On two things, the National Health and Nutrition Examination Survey is a wonderful database to examine for the first time the effects of multiple things on taste and smell, including medications, and it was really illuminated the income and health disparities, even with the sense of smell and taste. So there are lots of reports on medications influencing smell and taste, and so that has been reported, and it grows all the time. And so these medications can indirectly and directly affect taste and smell and flavor perception.
Yeah, yeah. We need to at least be collecting the data.
Absolutely.
We're not going to exclude.
Can I say something, Elizabeth?
Yes, please.
Going back to your question about socio-demographic, I have encountered some cultural differences and which have made a difference. Demographics-wise, I have mostly done studies in minority populations, so mostly there I've dealt with was more cultural and food differences, like their way of preparing food is different and the foods they eat are different. So those things I have dealt with.
Yeah, it was raised earlier today that being in a research study controls your food intake, right? And there's a certain amount of stress associated with not being able to choose what you're going to eat. And it makes me wonder if that impacts the results at all. I'd like to end with kind of a complicated concept. And this is that there are food components that might cause people to lose weight, like adding fiber to a diet, for instance, or lowering carbohydrates. And yet, when we try to keep people weight stable, my question is, are we overfeeding them? Because the diet we're feeding them is causing them to preferentially reduce their food intake. You understand what I'm saying? Yeah? So what kind of effect is there when you're keeping people weight stable when the diet you're feeding is actually inducing weight loss? You're feeding them more and more, aren't you?
I think I know what let me just kind of, so we run feeding studies as well, and we definitely find that when we give people, like when we actually feed them 100% of their needs, that they do gain weight. So I think, but at the same time, I guess it's probably slightly better quality than what they're getting. Like we definitely have more fruits and vegetables than what they're reporting eating, and that obviously has more fiber. But then at the same time, when we prescribe 100% of their needs, like during weight maintenance approaches, when they're on their own, they gain weight. So they usually end up eating more like 110%. So we definitely learned in the weight loss field, like you tell them to do 100, they'll do 115. So if you want them to actually do 25% restriction, you got to tell them to do like 40 or 35 or something. So I'm not sure if that's getting at what you're saying, but those are kind of patterns we've noticed.
It's the dichotomy between feeding a diet and then how well that matches their admin.
Mm-hmm.
Yeah. All right. Well, I would like to start to wrap up. Why don't we start from the top with Dr. Duffy and ask you to think about what the future of nutrition, precision nutrition, means to you.
Well, thanks again for the invitation. It's been a very exciting conference, and I think that the sensory nutrition field could benefit much from interacting with interventionists, and I think that the sensory nutrition can provide a lot to interventionists to think about, because the field of taste and smell is no longer forgotten. It's really, I think, at the forefront, and there's that exciting compendium that came out of the conference, not only for regulating or modifying what people choose to eat habitually, but also the exciting research that taste and smell receptors throughout the gut can have on the response to dietary intervention. So I think there's just really exciting collaborations that can go forward, and I highly, highly recommend, if you're going to do an intervention study, I gave you my top 5 list of things to consider, but I would highly recommend that interventionists reach out to sensory nutrition specialists.
Thank you so much. Dr. Barabási, have you got some final thoughts?
The different dimensions. Oops. Yes. So precision nutrition can have many different dimensions. I have spoken today about one aspect of it, which is actually having precise understanding of what are the chemicals that really reach us through our diets. But I think this is just one aspect of the story, in the sense that what I think is happening and what will happen in the coming years is that big data approaches would really kind of come and improve our ability to think about nutrition. And here I don't necessarily mean just simply what's in the food, but how do we eat? What do we eat? What is the reality really of our eating patterns? Is already being helped by the digital tools. And I think it will be actually transformative, particularly as people are recording more and more information about themselves. And I think this community needs to be prepared to take advantage of this digital revolution to kind of rethink the way we think about food intake, nutrition, and its health impacts.
Thank you. Dr. Voruganti.
I'm very optimistic, I guess. I envision someday when a person comes to a doctor or even a healthy person and they give us samples, we have a multidisciplinary team where metabolic people, genetics, epigenetics, microbiome, metabolism, and sensory, and other features. They all work together and come up with a plan for the person, preventative, and then tell them that you are genetically susceptible, you have to follow this dietary pattern. I know this may not be very near future, but I think we'll get there soon.
Thank you. Thank you. Krista. The future of your field.
Sorry?
The future of your field.
Yeah, I kind of touched upon it already, and it's just like what, is it Saroja, what she was just saying, I kind of just exactly where it'd be wonderful if we could, instead of just telling people to try whatever they want, if we could know who were the responders to certain diets based on genetics or microbiome or whatever it is, something measurable that we could pinpoint diets that work for them and then just give them a handful of diets to work with, opposed to them just like trying and failing at all the numerous diets out there. Because with that, there's a lot of diet fatigue, and then people just quit, right? They don't even want to do any of them anymore. So yeah, I think that's where the weight loss field hopefully is heading with precision nutrition.
Thank you. Erin.
I am super encouraged by workshops like this, and I thank you for the invitation and inviting us sleep scientists, because the bringing together nutrition and sleep and circadian timing, I think, is very important. Of course, I do as a sleep scientist, but I'm encouraged that others are thinking along the same lines. And I think just continuing to think about sleep and the individual's individuality in sleep need or the impact of sleep deficiency and time of day, just circadian timing, will be important for us to continue collaborating and having these conversations so that we can continue to move the field forward.
Thank you. And Dr. Malone.
I guess I'll piggyback on top of you there, Erin, and I'll say that we kind of know that rhythms are the rule, not the exception for most biological and physiological processes. So I really hope that in the future, we can advance our understanding of how rhythms and timing of lifestyle behaviors can increase not only the precision of our interventions, but also of our assessments for nutritional intake, physical activity, as well as for sleep.
Yeah, thank you very much. I would add, for me, it is that we tend to be relatively narrow in our focus, and what I've taken from today is that these, as many of you said, these transdisciplinary efforts need to be increased, and hopefully that will increase rigor in data collection. And to really figure out how we want to design studies and make conscious decisions. If you control the diet before someone's going to do an oral glucose tolerance test, you might get lower variability. But does that really, is that a good readout of what it would be like to do an OGTT if you pulled somebody out with no control? So control the pre-study diet, or don't control the pre-study diet, but figure out why you're doing it and make those decisions wisely. I think we should close here. I appreciate all of your time this afternoon and everyone online in participating in this. Thank you very much. Have a nice evening.
Thank you so much.